Okay, sound check for those at home. Somebody tell me whether you can hear me.
Can hear you.
Thank you, Tasha. Okay, well, let's get started today. So today's agenda is to talk about the evolutionary response to competition in ecological time. But before we do that, I had a couple of questions about the Wednesday's presentation. So I thought I'd revisit that just briefly. And so there are a couple of questions here, like on this analysis of competition between the invasive species, Albopictus, which is the dengue fever carrier, and Triceriatus, which is the native mosquito that doesn't carry dengue very effectively. And so the question was, well, how did they do this? And what they did was to do experimental arenas, kind of like we've been talking about, and they would add a certain number of larvae to the water in kind of an artificial tree hole. And they would then add some larvae of the competitor, and they'd see what the change would be. And so they were actually able to calculate competition coefficients. And because they did some of these experiments with only one species or only the other species, they were able to establish the carrying capacities. So if you know the carrying capacity and if you know the effect of one species on the other, then you can draw these lines. So if this is Albopictus here, what they first did was put Albopictus larvae in a little 100 milliliter cup and ask how many can that support. Turns out it can support about 55, 56 of them right here, because that's K for Albopictus. And then they looked at the number of Triceriatus that could be supported in 100 meters, 100 milliliters in an artificial tree hole, and they found that it was about, what, 43 maybe, something like that. But then they calculated the effect of each species on another by saying, how much is the carrying capacity reduced if we put in 10 of the competing species? And so, obviously the numbers would drop. And one of the questions I got was, well, what happens if it's not the target species that drops, but the species that you added? Say you added 10 and a couple of them die. Well, that's why you have to do experiments, because you put those 2 back in, put 2 new ones to replace the ones that died, so you maintain the density at 10. So if you maintain a constant density of competitors at 10, you can then ask, well, what can that environment support for the other species? If it could normally support 56 or so, 55, 56 albopictus, how far down does that go when you add 10 individuals of Triceriatus? Well, you take that number divided by 10, and that's the effect of a single Triceriatus on the carrying capacity of albopictus. And what you can see here is that when we divided K by alpha, which is the effect of Triceriatus on albopictus, it's less than 1. Because here it's 55, and over here on the other end, 55 divided by the effect is about 75. So probably it's somewhere around what? It's probably somewhere around 0.6, 0.7 maybe. So each individual of Triceriatus reduces the carrying capacity of albopictus by about 0.6 or 0.7. That's how you get a bigger number here. Okay, and it's important to realize that this then indicates it's a stable interaction. It's a stable equilibrium right here. So they did the same thing for Triceriatus. They said, okay, if we get 43 Triceriatus at carrying capacity in this little experimental arena, and we add 10 albopictus, how much does it reduce the population of Triceriatus? And so what you can see here is that 43 divided by whatever beta is quite a large number. So this is about 0.5. It's actually a bit less than 0.5, probably about between 0.4 and 0.5. So each individual of albopictus reduced the carrying capacity of Triceriatus by about 0.4 to 0.5 individuals. But that's how they did it. They took, say, 10 larvae of the other species, made sure that they kept 10 larvae in there. And then they said, well, what's the stable K for our target species? And that's how they were able to do this. Okay, and what you can see over here in tires, kind of, again, these are not real tires laying around on the highway in Florida, although that's kind of an attractive visualization there. It's fake tires, and again, it's standardized by 100 milliliters of fluid. And what you can see is that the Triceriatus isocline is not terribly, terribly different over here, but it's quite different here. This effect of albopictus on Triceriatus is really, really much larger. It's about 0.9. It's about twice the effect. And over here, you can see that because albopictus isocline goes way up there and intersects at about 200. The carrying capacity of albopictus is reduced almost not at all by Triceriatus, which is why albopictus can drive Triceriatus extinct. There's this huge, huge mismatch between the effect of the two species on one another. Albopictus has a large effect on Triceriatus, especially over here, but Triceriatus has almost no effect on albopictus. It takes 200 Triceriatus to use the same amount of resources that about 45 Albopictus would use. So there you go. They have almost no effect on the invasive species when they're nesting, when they're developing in tires. And why that is, I have no idea. Okay, a couple of other questions came up about the assumptions of using these things. And so I want to quickly run through those. Remember, we have 3 assumptions that are really required for us to use this to predict things. We can always use it to calculate what the interaction is and describe a given interaction if we know what the stable numbers are for 2 species. But can we use that to then predict something else? Well, in the example of the mosquitoes that we just had, yes, we did. We were able to use our calculations where we described what happened in those experimental chambers. We were able to predict what happened in nature, and it was pretty darn close to what actually happened in nature. On the other hand, you have to have 3 things be true for that to happen. The competition coefficients must be constant, regardless of density. And they have to be constant for a given interaction, and they can't change if the environment changes. And that's kind of the key to the previous experiment, was that they didn't make the assumption that what they found in a glass beaker would be exactly the same as what they found in a tire. So they did separate experiments to be sure that they didn't make this assumption and have it bite them. And the third thing is that in real communities, 2 species don't interact alone. They interact in the presence of others. And some of them may be there, some of them may not. And so you want alpha and beta to be constant when other species are added. All right, so this first one then shows that the equilibrium between 2 competing species is not where we expect it to be. So based on our calculations here, we were able to calculate the carrying capacities right here and right here, and the endpoints of the isoclines here and here. That should predict stable coexistence right about here. But instead, stable coexistence did occur, but way over here. So that was wrong. So the question was then, well, how could we make such a massive error? And it turns out that when you do the experiment, but you carefully control the number of competing individuals so that you have 1 competitor in a chamber, and then 10 competitors, and then 20 competitors, and then 30. The lines are not straight. Adding a single competitor of another species right here, so adding serrata, adding 1 individual of serrata causes a big drop in the number of pseudo-obscura. And that drop tends to lessen as you add more and more serrata. So it's not a linear thing. The alphas and betas are not constant with density. The first couple of competitors cause a big change. Adding additional competitors doesn't cause the same degree of change. It causes less and less change. And that's true for both of them. So adding a single individual of pseudo-obscura here causes the number of serrata to drop pretty dramatically. So from here to like here, that's a really dramatic drop. But then you add more and the drop is less. So that's the nonlinear thing. And once again, the way we found this out was by doing these experiments where we control the number of competing individuals and say, okay, how much is K reduced when we maintain a constant number of 10 or 20 or 50 competing individuals? Okay, for this one, once again, and the questions are all on how do we calculate these things? Well, once again, what you see here is that this is the equilibrium population size in competition, but it's not the carrying capacity, right? The carrying capacity was established separately with these populations growing in isolation with no competitors from another species. But the point of this is that the average equilibrium population here of 26 versus 2.6, what that means is that there were many instances in which this species was extinct. Okay, and because ecological experiments are noisy, they did 20 to 30 contests at each of these combinations. So maybe out of the 30, maybe 28 times this one went extinct. And the remainder had a few individuals, and so the average was like 2.6. Okay, so these are the averages of many experiments. Okay, finally, went through this one a little bit fast, and I just showed you that we had two sets of kind of model communities of microcrustaceans, and there were four of them. And in one community, this fourth species was present, and in the other, the fourth species was absent right here. So the question was, if you calculate the competition coefficients, do they stay the same? Is this effect the same as this effect? And it's not. So what we do is ask what the equilibrium population is at carrying capacity, and that's 614 for this one. So for Allanella, there's 614 Allanellas if there's no competition from another species. And if you add individuals of this other species, Seriadaphnia, it drops down to 417. So there's almost a, it's a difference of almost a third. So the carrying capacity is reduced by about 200 individuals. How many individuals of Seriadaphnia did that take? Right around 300. So 200 divided by 300, that's the competition coefficient, about 2/3. So that's how we got those calculations and how then we can compare them. Because without Hyalella, this guy, Allanella, has a huge population, has 6 times the number of individuals, right? With Hyalella, it only had 614, but without it, it had 3,600. So clearly, they're competing for resources. But now, if you add Seriadaphnia, it drops to 2,280. Okay, and it took 900 Seriadaphnias to do that. So this drop of 1,400 divided by the 900 that it took to produce the drop gives you this competition coefficient. And very clearly, 1.47 is not equal to 0.67. That's a pretty huge difference. So what we're saying is that these are things we can understand when we have two species competing. But we have to be very careful about applying this to try to predict things in more complicated situations. We have to actually put in the groundwork to determine whether alpha and beta are linear as density changes, or is there an effect of density? Does the first one have a much greater effect than the 100th one? Or maybe the first one has virtually no effect on the population carrying capacity. And it takes a whole bunch of them to start reducing carrying capacity. Well, that would be nonlinear also. So we have to do enough experiments to understand that. Okay, we also have to be sure that we're comparing apples to apples and that the environments we're comparing are not so dissimilar that your calculations can't translate. And finally, we have to be very, very careful when we try to predict what's happening in more complicated communities, like actual communities. So this is a very helpful thing for us to do when we're dealing with two species and when we have some experimental control. But if you're looking at real communities where you don't have experimental control, it can be really tough to generalize from these two species interactions. Okay, so I hope that's thorough enough to answer those questions. And I'm going to stop sharing this one. And I'm going to get rid of that. And I'm going to share the other one that we're meant to share today, which is a bit of a briefer presentation anyway. So before we continue on, I'd like to ask if anybody has questions about especially how you calculate those competition coefficients. It's really not hard. Okay, well, so what we've done is looked at competition in ecological time. That is, we've looked at it over a very short time period, and we've asked, what kinds of patterns can we see? Well, we can see that in ecological time, if you try to put two competing species together, a couple of things could happen. One of them could drive the other extinct. They could coexist stably, or they could kind of coexist unstably if you kind of get the numbers right. But anything that perturbs that equilibrium will cause one of them to drive the other extinct. So that's in ecological time. That's the short term. That's how we know, for instance, that two species that can coexist with overlapping niches will exclude each other from a part of their fundamental niche. But if we allow that to go on over a much longer period of time, over evolutionary time, where you have multiple generations experiencing that kind of environment where you're competing for some of your resources, what is the response? Well, the response is called character displacement. It's where the characteristics of species diverge to reduce the intensity of competition. Again, these characteristics can be physical, they can be biochemical, they can be behavioral. If you have two species that are diurnal and they're competing for many of the same resources, you may get one of those species becoming more and more crepuscular, that is morning and evening, or even switching to nocturnal. Well, in that case, they would be trying to use the same kinds of resources, but not at the same time. So the degree to which their niches overlap would be reduced through natural selection. So what we're going to do is try to figure out how natural selection proceeds to cause the characteristics of these species to change over time. All right, so here's a cartoon where we have high niche overlap. The likelihood of two species sharing this amount of their niche is that very likely one of the species could drive the other extinct. But for the sake of argument, let's just say that it's just enough to allow them to coexist. So now we've got individuals that are coexisting, and let's look at the competitive environment that the individuals experience. So you have some individuals of this species on the left that would like to use resources in this part of the axis. This could be salinity, could be whatever. You have other individuals that are using resources in this part of the axis. Okay, and what I want you to understand is if you are using shared resources that another species is trying to use, you're experiencing extra competition for those resources. You're competing with your own species for these resources, and you're competing with the other species. So these are going to be less available. You will be more resource limited if you're trying to use this set of resources than if you're using this set of resources. If you're willing to use these resources, you have them all to yourself. You're only competing with others of your species. That's a lot less competition. Okay, and now we're going to take the next logical step and ask, well, how does the fitness of these individuals differ? Well, if you have these resources all to yourself and you're only competing with others of your species, you have access to a lot of energy. Let's just say that these are energy, these resources are energy based. On the other hand, if you have to compete with your species and another species, you're going to get a lot less energy. All right, so let's think back to the principle of allocation. Energy can be used for one purpose and not two purposes simultaneously. So if you want to survive in use these resources, how much energy can you devote to reproduction compared to individuals that are surviving and using these resources, where they get more? Very clearly, if you're using this set of resources, you will have a fitness advantage. You will reproduce more than individuals that are trying to use these resources. So what is the outcome? There will be a shift in resource use away from shared resources, which are really limited, because the individuals that are trying to use the shared resources will reproduce less than the ones that are using the exclusive resources here and here. These guys will have higher fitness than their conspecifics that are using these resources. These individuals will have higher fitness than their conspecifics that are using these resources. So there will be a shift in resource use over evolutionary time, because whatever characters that allow you to use the extremes will give you higher fitness. So that's going to cause resource use to shift from the middle to the extremes. And that's what it's going to look like after evolution acts on this interaction. What's happening here is not displacement of resource use. It's displacement of the characteristics of the species that allow them to use these resources. So this might be seed size. What character displacement does is change your mouth so that you are more efficient at using small seeds versus large seeds. Okay, so character displacement is not about the resource. It is about the features of the organism that allows them to use different kinds of resources. If you're trying to use, say, a salinity gradient, character displacement would change kidney function for those that are on the saline part of the gradient, and it would change kidney function in the opposite way for those that are on the freshwater end. What's changing is the characteristic of the species. It's not the resource itself. That's an important point. Yeah, question here. Yes. Okay, so the question is, this changes the characteristics of species and helps them adapt. The answer is yes. And the key thing to understand here is that remember that the niche is a collection of tolerances and requirements, right? Well, some of that is based on the physical environment, just how hot can you take it? But some of it is based on the biotic environment. Who do you have to compete with for your resources? So it's both the physical environment, which is easy to grasp, and the biotic environment. And that's a little harder to grasp. And that's why we're spending an entire lecture talking about the biotic environment shaping the niche. Now, if you think about it, this distribution of resource use defines the niche of these two species. So the niche has changed. It has changed because their physical features have changed. Their traits have changed. The resources haven't changed, it's the traits of the species. Okay, so in the situation on the right then, most of the individuals of each of the two species are using resources that they have exclusively to themselves, and they have higher fitness than individuals that were trying to share this set of resources here. The reason that it's changed is because these guys that are using the extremes here have higher fitness. And so over time, the traits of these two species will diverge. Let's say this is beak size that defines the niche, and here's the seeds that they're using. Well, the ones with the smaller beaks that use the smaller seeds have higher fitness than the ones that have medium beaks. And here, the ones with larger beaks that allow them to use the largest seeds have higher fitness than the ones that are sharing. So over time, if the small ones out reproduce the large ones, this is going to move away from the center. If the large ones here out reproduce the middle ones, this utilization curve will move out. Okay, so what has to happen then is that these species must coexist long enough for evolution to start shaping them. Once evolution does that, it changes their niches. And that's why we think that competition is actually a very pervasive thing in nature. We don't often see it in action as much as we might expect. But what we see is a lot of instances where we have two species that look very much like this. And the inference is that in the past, they may have looked like this. And in some cases, we do in fact see this happening. All right, so this is, as I said, really difficult to see in action in nature. So we have a number of natural experiments in which we think that we understand how character displacement has worked. And here's probably the best known. This is beak morphology of the Galapagos finches. So if you remember anything about evolution, you'll remember that one of the things that shaped Darwin's thinking was visiting the Galapagos, finding that there were all these finches on the different islands, and realizing that each island had a different bird. And what you frequently don't get told is that Darwin didn't really understand how significant this was at the time. So he just went out there with a shotgun, shot a bunch of birds on each island, then threw them all in the chest without sorting them very well. And what it took was him going back to England and talking to someone who knew something about birds before he realized, oh my God, I completely messed this up. If we sort them out, look at what we find. So here is an example of what we believe to be character displacement. So here's the Galapagos, and we have a bunch of islands. Some of them are big, some of them are small. The big ones can support more than one bird species. The little ones can only support one. So let's look at beak size, and there's a reason why I talked about beak size earlier. Let's look at the beak size of 3 different finches on these islands. Okay, so we're going to look at Fuliginosa, which has a kind of a narrow beak, not narrow side to side, it's narrow up to down. We have Fortis, which has a medium-sized beak, and we have Magnorostris, which has a really deep beak. Okay, these beak sizes enable these birds to use seeds of different size. So Magnorostris, with its enormous beak, would be very inefficient trying to pick out small seeds out of the soil. But Fuliginosa can do that very easily. On the other hand, Fuliginosa cannot do anything with large seeds because it doesn't have the strength to crack them. Magnorostris has this big, robust beak, and it can eat those large seeds very easily. So these guys eat the largest seeds. They're better at getting them, and they can crack them. These guys eat the smallest seeds. They're better at finding them and handling them, and they can't deal with the large seeds. And Fortis is somewhere in the middle. So let's look at the patterns of their beaks on islands where they do and they do not co-occur. So on the larger islands, Pinta and Marchena, this is the distribution of beak depths for Fuliginosa, for Fortis, and for Magnorostris. And notice that these are quite large, these are medium, and these are small. What happens if we remove Magnorostris? Well, we can't do that. That's illegal. Ecuador doesn't like it when you kill their birds. But we can go to neighboring islands that are a little bit smaller, Floriana and San Cristobal, and ask, what's the distribution of beak sizes? And what we see is that there's not a whole lot of change in Fuliginosa here. There's still a peak in Fortis at about the same size. But look at the tail. Look at the tail of this distribution. Some individuals of Fortis are able to be much bigger than they are on the islands where Magnorostris occurs. These guys are able to use the large seeds because their beaks are so much larger. Okay, and the inference then is that, well, at the extremes of their distributions here, they're competing for the same seeds. When you remove the larger species, then some of these individuals with larger sizes can get enough food. And character displacement then is responsible for this difference. If this is the natural, quote unquote, distribution here of the maximum beak sizes, so if the tail naturally would look like this, and the presence of a larger competitor causes that tail to be truncated right here, that's character displacement. The individuals that might have lived on these 2 islands that would have very large beaks can't get enough food because of competition from Magnorostris, and so they die out. The ones with the smaller beaks dominate. So the character displacement has occurred here. Fortis's beak size has been reduced by competition with the larger species. Let's then look at islands where we have only one of the 3 species. Let's look at the 2 smaller bird species. Okay, so here's Daphne, which is a very small island, and it only has the medium-sized Fortis. Okay, well, look at where the mean beak size is. It's much smaller than it is here. And the interesting thing is, if you look at Fuliginosa, the smallest one, look at where its distribution of beak sizes is. They are identical. These guys have identical beak sizes when they occur individually. And where they occur together, Fuliginosa is displaced to smaller beaks. Fortis is displaced to larger beaks. We know that this is very consistent on the several islands in which we see this. Fuliginosa always has the smaller beak where they co-occur. Fortis always has the larger beak where they co-occur. And that's because if you look at the lowest size of Fuliginosa beaks, they're a lot smaller than the smallest size of Fortis beaks. If you look at the largest size of Fortis beaks, they're always a lot larger than the largest size of Fuliginosa beaks. So there's kind of a bias. Fuliginosa is biased to evolve smaller beaks when it's in competition with these species. Fortis is biased to evolve the larger beaks. Now, there's a problem with this. We don't know exactly how this happened. We think we know. But there are many ways in which these islands differ from one another. It's not just the number of competitors on the island. So we don't have the same kind of control we'd have in an experiment. And so this suggests that character displacement has occurred. But since we didn't actually see it happen in ecological time, we don't actually know that it occurred this way. It's pretty darn consistent. And the more consistent we see it, the more confidence we have. But as scientists, we have to understand that this is not proof. This is suggestive. It's consistent with character displacement. And when we look at the 20 or 30 islands in the Galapagos, every one of them shows similar patterns. So we're pretty confident, but it's not proof. It's not an experiment. We did not control absolutely everything about this interaction over evolutionary time. So we cannot say for sure that this proves that character displacement has occurred and is responsible. We can say that it is consistent and that the evidence is extremely strong, but we can't say prove. Need to be clear on that. And this is a problem with ecological work in general. We have less control than in the laboratory sciences of biology. So in the laboratory sciences, you can do a titration over and over and over again. You can control all of the variables except one. You can be absolutely certain. We almost never are able to have that level of certainty in these ecological patterns because we don't have the same level of control. So it's consistent. The evidence is extremely strong. We are highly confident, but we can't say it is because. We can't say it proves it. All right, so finishing this off, let's look at what happens to the competition graphs when we think about the process of character displacement. Well, let's look at a graph that might represent this interaction where you have high overlap. Okay, so you got a lot of overlap here. The effect of one species on the other is probably pretty high. Okay, in that case, then this is a situation in which we might expect the unstable coexistence case. And in unstable coexistence, the carrying capacities are out here, and the ends of the isocline are inside. So in this situation, if you have, say, 50 here and 50 here, and this is less than 50 and this is less than 50, then obviously you're dividing 50 by a number that's greater than 1. So this would be very strong competition. the effect of one species on another would be greater than adding another individual of that species. Okay. This is unstable competition. What happens as the characters of these species are displaced through evolution? Well, they move apart, and look at the level of overlap. The level of overlap decreases dramatically. And that means that adding another individual of the competing species is going to have less and less and less effect on the other species over time. So if you divide the carrying capacity here by the competition coefficient, this competition coefficient is large, it's greater than 1. But what we're saying is that over evolutionary time, that competition coefficient gets smaller and smaller and smaller until it drops below 1, might drop substantially below 1. And now you'd be dividing 50 by a number less than 1. So the ends of these isoclines would gradually move out this way. And they'd move out this way over evolutionary time. As the competition coefficient gets smaller and smaller, this greater than 1 here, still greater than 1, but not as much. And now it's exactly 1. But if it moves that way, the only way that can happen is that if the competition coefficient is less than 1. Well, that means that the Ks will be on the inside. And you'll have switched from an unstable interaction to stable coexistence. And since we see so many species coexisting stably with competition coefficients less than 1, and yet they're quite similar species, we think that this process has probably been really common over evolutionary time as new species emerge. And that competition for shared resources is one of the major drivers of speciation. As characters are displaced, who are you going to mate with? You're going to mate with somebody who has the old character where your offspring have to share resources? You're going to mate with somebody who has the new character that allows your offspring to have all the resources to themselves. It's not surprising who's going to win that. So stable coexistence, we think, has been generated by character displacement in evolutionary time in a huge number of ecological interactions. So that's why there's this emphasis on understanding these competition coefficients, because it helps you understand how these interactions work and how they will change over time. And that's why I kind of insist that I want you to be able to calculate them. So there's a couple of problem sets on Moodle now on competition. There's also that problem set that I promised you on population matrices, about translating a life cycle into a matrix, and about calculating the number in the next generation based on the matrix. Be sure you do those. Those are important. And that's all we've got for today. So, do we have any questions, either here or at home? Yes, question here. Okay, did Darwin actually discuss character displacement in The Origin of Species, or did it come later? He got back from his voyage on the Beagle in the 1830s. He didn't publish The Origin of Species until 1859. So he had a lot of time to work through this. And yes, it was included in The Origin of Species. He had a good 20 years to work through the idea. So it wasn't a bolt of lightning that inspired him. It was careful, persistent work, staring at his specimens and trying to understand the patterns that he was seeing. For Alfred Russel Wallace, it was a bolt of lightning. Wallace was the other guy who came up with the idea of natural selection at about the same time, but he conceived of it during a bout of malaria when he had a high fever and was in his hammock, probably hiding from the Malay pirates who were trying to kill him. So, but Wallace had the idea, Darwin had the data. That's why we talk about Darwin more. Other questions? Okay, that's all for today then. I will see those of you who are in lab in about an hour, and we'll have some fun with duckweed and wheat. And in the meantime, I will end this for us all and get this posted as soon as I can. And the exam will be back to you on Monday. So try to decide whether you want to come to class and pick it up in person or what. And goodbye to all.